Excellent. Well, it's a great pleasure to be presenting today, and particularly to present with two of my students and who will be able to represent work going on in the lab and what the projects they're driving. So I'm going to be talking mainly on ovarian cancer today, and really focusing on the way we're thinking about the disease and how that is obviously it impacts the way we approach our research at this point in time. So I'd like to start with a dedication. About 20 years ago, my mentor in pathology, Blake Jilks, introduced me to Diane Miller, and Diane created this incredible opportunity to make a difference in the world, which was OVCare. And I'd like to thank them both for the profound impact they've had on my career and the opportunity they created, which all of us who work in gynecologic cancer research in BC appreciate and benefit from. OVCare was very focused on ovarian cancer, and when we started, but we realized you can't just study ovarian cancer in isolation. And so we did something very British Columbian. We decided to make our team bigger and more inclusive. And so we joined up with researchers studying cancer of the cervix and created what we call the Gyne Cancer Initiative. And we were motivated by a very ambitious goal of can we pull together all of our expertise and our efforts, strategically recruit and push what we're doing out into the community and decrease death and suffering from these cancers by 50%. And we do this by a combination of biologically informed prevention strategies, biologically informed diagnostics and treatments, and biologically and social culturally informed survivorship strategies. My research really falls mainly into the first two. This is obviously really big, and a team focused on my own laboratory could not even think about addressing something so big. I don't have the expertise. And so we really had to expand our team. And we're really fortunate that we've managed to grow our team to include many excellent researchers who've increased our grant funding base and increased our capacity to address our purpose, which is the 50% reduction in death and suffering from these cancers. I should note that all of these PIs are supported by and privileged to work with even more talented and hardworking trainees, and they're the ones who do the work. So when I started thinking about ovarian cancer was really when I joined the pathology department as a resident. And Phil Clement, who's a world expert in traditional pathology of gynae cancers, taught us to what these cancers look like. And they're really interesting patterns. Some of them were connected to specific clinical scenarios. But that was really as far as it went at that point in time. Indeed, even into the last decade, there were points of view that basically these cancers are all basically the same. They should be treated the same way. So it doesn't even matter what you call the tumor or what you understand about it. It was a very defeatist attitude. And that really did contribute to the fact there were no real improvements in ovarian cancer, and a much used figure where we contributed data. And as generated by Gillian recently, we can see there actually hasn't been a huge shift recently. So it's a disease we must do much better at. The first thing we did as OVCare is to prove that these are absolutely distinct diseases, basically every way you can define a distinct disease. And that's important because then you can have a stratified approach to prevention, treatment, and you hope at some point survivorship. And so we impacted the WHO classification, which simplified things, and we proved that community pathologists can make this diagnosed with 95% accuracy. And unlike in breast cancer or in uterine cancer, molecular analysis falls under the histologic classification as opposed to competing with or replacing. So these are distinct diseases of every way you can define a distinct disease. So our premise shifted to if these are distinct diseases, let's study them this way, let's treat them this way. And this is kind of an interesting shift. Right now, it's just the way people work and think about ovarian cancer. But when we first started pushing this forward, it was violently opposed by a lot, including research-focused oncologists who felt that this would make their trials more difficult. But I think we prevailed. So we say each histotype is a different disease, needs distinct solutions. It means success in one subtype doesn't mean something's going to succeed in another, because they're different problems, i.e. platinum taxane may work a bit in high-grade serous cancer of the ovary, much less in clear cell. But that also means failures are not transferable. Just because HER2 therapy didn't work in high-grade serous cancer, it doesn't mean it won't work in HER2-amplified mucinous cancers. And ultimately, what it comes down to is we need to study these diseases as specific entities, and it means we do need model systems which are predictive of the behavior of these biologic entities. So once we determined this, we embraced an opportunity to sequence these tumors in collaboration with the Genome Sciences Center. And we got very proud of ourselves because we made discoveries of the key mutations in several types of ovarian cancer. And indeed, we were somewhat filled with hubris about 10 years ago. And that we thought, wow, okay, we've solved these cancers. We found a mutation which defines them. But it obviously just isn't that easy. So our thinking at this time would be each subtype would be defined by distinct mutations. And this fit very well into sort of the paradigm of precision oncology, which still is very important in oncology, and where you could take a disease, lung cancer, split it up into a bunch of bite-sized groups, all of which have different treatments. Now, this actually worked curiously well for lung cancer, but not nearly as well in other disease spaces. So precision oncology, panel testing for common features readily available, larger scale genomic sequencing becoming available for genomic signature assessment, and we're starting to do this with low-pass whole genome sequencing in the endometrial cancer space. Genomic classification of endometrial and other cancers has moved into the clinic. And we use ctDNA testing as an adjunct diagnostic in many clinical scenarios. And I'm heavily invested in this space. Our company Connexia does work in this, declare a conflict of interest, but it is important, it's made a difference, but it doesn't hold all the keys to the castle. Because finding a mutation doesn't take you to a treatment. In some cases, it may take you to a diagnostic, but it's because it doesn't tell you enough about the disease to move forward. Why so difficult? Well, mutations targetable in one cancer type may not be targetable in another. So BRAF targetable in melanoma, not in colon. Combinations of events, which could be synthetic lethal interactions in one scenario, may not be lethal in another, or may even define the cancers. So SMARCA2 and A4 deficiency in combination or a synthetic lethal interaction in lung cancer, but small cell hypercalcemic ovarian cancers and some D-diff carcinomas have that event, it defines it. And so treatment approaches will not be transferable. Some cancer mutations are even more common in non-cancerous tissues, which is problematic. And really few of the major new mutation types which were discovered by large-scale sequencing have been effectively targeted. So there is a sort of tendency that when you do something well and you did some good, you just keep doing it until people say, no, stop. And so there's these efforts to do more and more sequencing of tumors to discover more things, but we actually haven't figured out what to do with the major mutations which we've discovered already. And I would think that finding out what to do with a mutation present in 50% of a cancer type is more important than finding new mutations present at less than 1% frequency. And so the main mutations, mutation signatures, and combinations of mutations have been discovered already. The implementation of precision oncology into the clinics is challenging. Most patients still don't get the tests they need when they need them. And of course, in the same way, this absolutely horrid loaf of bread cannot be explained by a bad recipe. The genome itself does not explain everything going on in a cancer and what has gone wrong with the cells which make up that cancer. So we had to think about cancer differently. So my thinking now, and this is what influences all the work in our team, is that each cancer type is an endpoint of a distinct oncogenic journey. And this is a journey I took last summer. It was a walk from hope to Tulamine I did with Blake Jokes. And like any journey, where you end up and in what kind of shape you are in and what you look like is dependent on where you start, the terrain you cross, and what happens along the way. So here you can see we saw some beautiful things, but I also had to eat Blake's favorite camping food, which is fried spam, but I survived it. And here's us ending up in Tulameen at Diane Miller's cabin. So when we think of an oncogenic journey, so every cancer we see isn't a random uh, entity. These are very specific entities which represent very specific oncogenic journeys. In other words, there aren't infinite ways to make cancers. There's very specific ways to make cancer. And the endpoints are definable. That's why we have histotypes. So what makes up an oncogenic journey? There's a cell of origin. There's mutations, what happens along the way. And then there's the microenvironment, the terrain you cross. In our thinking now, all of these have to be for if we're going to understand these entities and make a difference. This is particularly true of mutations which impact the epigenome, where you can't tease apart these features. So cell context is defined and maintained by the epigenome, therefore the impact of mutations and drugs that perturb the epigenome will always be cell context specific. It has to be. So we cannot think about mutations in isolation. And so our hypothesis, which really covers all of the work in the team, is understanding these oncogenic journeys in terms of cellular origins, mutations, and microenvironment will lead to prevention and treatment opportunities. Now, rare cancers and common cancers are a little different in how these oncogenic journeys map out. And I like to use an analogy of a luge run compared to a ski hill. So rare cancers like a luge run, where you tend to have a very specific cell of origin and single dominant pathogenetic pathway, which can lead to, if you understand that pathway, a dominant treatment which will work for all. Whereas common cancers, let's talk about like lung cancer, it's more like a ski hill, where you have a cell of origin, which may be the same, you have an endpoint at the bottom of the hill, which is the same, but you have a whole bunch of different ways from getting to A to B. And these distinct ways may be targetable using a precision oncology approach. So I'm going to be using rare and common cancers to kind of think more about this cell context mutation microenvironment kind of concept. So we study several rare cancers in our team, and adult-type granulosa cell tumor, DICER, and small cell hypercalcemic, where we found the key mutations. I won't be talking about DICER, but I would like to note that through much work done led by Yemen Wang and the team with Shari in the Animal Center, we've actually, and through our collaboration with Greg Moran, who's on the call, we now actually have a DICER mouse, which is producing the first DICER mutated tumors, which are adenosarcomas, and we hope to start making some Sertoli-Leydig cell tumors. But I'll start with granulosa cell tumor of the ovary and what we've learned. So this is about 5% of ovarian cancers, protracted course, known to be derived from granulosa cells. So we found this key mutation. It was our first discovery in next generation sequencing, and actually the first clinically useful discovery from that whole suite of technologies. But what we find this mutation, and it is a diagnostics, it's almost only found in that cancer type. So it's got about a 95% sensitivity and a nearly 100% specificity. And so why do we only find it in this cancer type? Well, it turns out that in the adult, ovarian stromal cells are one of the only places where this gene is expressed. You also see it in some stromal cells in uterus and fallopian tube, but curiously, we don't get the same mutation there. So the mutation must also tie into the specific biology of granulosa cells. In fact, likely what it does, it prevents the cells from dying. Because it's not a non-proliferative tumor, but how this happens isn't really understood. So now we're over 10 years since this discovery of the mutation was made. And only last year did some real insights come out in terms of what the mutation may do. So there were two papers, one led by Martin Hurst with Greg Moran and others, which showed that FOXL2 impacts DNA binding specificity of the mutation of this transcription factor, which makes sense. It's a transcription factor. That's the most linear way you could think that a mutation could be pathogenic. And there was another paper which had a somewhat different conclusion, but in the same space, which has suggested that this mutation changes binding partners and for the transcription factor, and that's what opens up the potential to lead to the transcription of a range of other genes and oncogenesis. These are both very credible papers. Another paper came out at the same time, which suggested that the mutation actually leads to a alternative microRNA site and degradation of message, suggesting that the mutation is sort of a tumor suppressor mutation. And Jessica Pillsworth, who's been working very hard on this disease, did a nice analysis which showed that this just can't be true, because if the mutation led to degradation of the RNA message, if you looked at tumors, you would see less mutant than wild type message. And here we can see in red the mutant message, and in blue the wild type message in a couple of normal cells without mutation at one end, and then in some heterozygous tumors. Now, of course, normal cell contaminants decrease the mutant threshold, but if you look at the homozygous or hemizygous tumors at the end, they're expressing lots of message. And we see this in protein as well. The tumors which appear to be homozygous, they only have the mutant message, they express lots of FOXL2. And so it's not really credible to suggest that this microRNA pathway could be the root of the pathogenesis of this mutation. So where are we after 10 years? Well, we defined the disease and we have a diagnostic. We still have no biologically informed treatments. And the reason why this has been so painful is there's a complete lack of model systems. And Jessica's been working incredibly hard and is making great progress in developing new models to study this cancer. Small cell hypercalcemic ovarian cancer was first discovered by Scully and colleagues in 1979. And he was sort of the grandfather of gynecologic pathology. A rare cancer, we get 1 or maybe 2 every year in BC. And it's a horrifically aggressive tumor. With about a 35% 2-year survival. We and 3 other groups found mutations in SMARCA4. And the mutations have a classic tumor suppressor gene pattern of a mixture of truncating mutations, splice site mutations, and some of them are actually germline. It leads to loss of expression of protein. And we showed how we could use this as a diagnostic in a very lovely paper led by Trainee. And the nice thing about it is it's an easy antibody to use, and there's a lovely internal positive control because normal cells express the SMARCA4 protein, which is called BRG1. We got excited because in this complex, the switch-NIF complex where BRG1 sits, there's an alternative protein called SMARCA2. And there were papers suggesting that if you're missing BRG1, you can knock out this other one and the cells would die. But we were quite surprised when we looked at this tumor and found that the cells don't express this other protein either. So it's a combination of features which would be lethal in almost any other cell type actually defines this cancer. And now at this point, I'd like to pass the microphone on to Jen G, who's going to talk about a specific project she did in this cancer type, which I think will be interesting and is very innovative.
Thank you, David. Thank you for giving me this opportunity to present this work. So next slide, please. And as David has already mentioned, so each subtype of ovarian cancer are really different diseases. But however, despite these molecular and clinical differences, these different subtypes of ovarian cancer are largely still getting treated similarly with conventional taxane and platinum-based chemotherapy. So this study focused on surveying some of the more non-high-grade serous ovarian cancers and to see whether or not we can find novel therapeutic targets for them. My apologies for the baby. Sorry, can you go back to the previous slide? Sorry, as David's already mentioned, so one of these. Okay, please, thank you. Sorry about that. One of these uncommon subtypes is small cell ovarian cancer hypercalcemic type. While the majority of the epithelial ovarian cancer types are diagnosed at older in older patients, the median age of diagnosis for small cell ovarian cancer, or SCOOT, is 24 years old. And as the Kaplan-Meier plot shows on the bottom, most patients succumb to their disease within 24 months of diagnosis. So there was a study in 2007 which looked at 24 of these patients and treated them with very aggressive chemotherapy, 4 chemotherapy times 6 cycles. And of these patients, the median survival in 3 years, 3-year survival is only 49%. So next slide, please. So then we asked, can the protein landscape help identify some therapeutic targets for these non-high-grade serous and uncommon ovarian cancers? So to do this, we used SP3 clinical tissue proteomics developed by Dr. Greg Morin's group in the GSC. This allows us to use archival tumor tissue that has been formalin fixed and embedded in paraffin already. So we compared some uncommon ovarian cancer subtypes, which include clear cell, endometrioid, and SCOOT, to the most common subtype, high-grade serous ovarian cancer. Next slide, please. So as shown here, we found that argininosuccinate synthase is at a lower abundance in some of these uncommon ovarian cancers, including SCOOT, compared to high-grade serous ovarian cancer, shown by this volcano plot here. ASS1 is labeled on the plot, showing that it has a significant p-value and a log2 fold change of about 2.5, which means in high-grade serous, there's about 5 to 6 times more of this protein compared to SCOOT. So next slide, please. So what is argininosuccinate synthase, or ASS1? It's a essential enzyme in the cellular synthesis of arginine. So arginine is a conditionally essential amino acid, which means usually we eat enough of it, but when we have a deficiency, cells can turn on this urea cycle and make arginine from citrulline, therefore sustaining cell survival. So as you can imagine, cancers that are deficient in this enzyme are more susceptible to its deprivation. So it is found to be highly expressed in high-grade serous cancers, and that in a smaller cohort of study, Chang et al. Showed that clear cell and endometrioid have lower expression of this protein, and that cell lines that are deficient in ASS1 are sensitive to ADPEC treatment. Next slide, please. So ADPEC is arginine deaminase, which is a bacterial enzyme that metabolizes arginine directly into citrulline. Next slide, please. So as you can imagine, when cancers are missing ASS1, they cannot complete this urea cycle. Therefore, they're really missing arginine for their protein synthesis, and they cannot really handle the deprivation of this amino acid. So this agent has been studied in phase 2 and 3 clinical trials in a number of different cancers, but ovarian cancer hasn't had a trial before, probably due to the fact that most high-grade serous cancers express high levels of this protein. So next, please. So as we mentioned before, our proteomic study points that some uncommon subtypes have lower expression of this protein, but we only had 6 cases each. So we expanded our cohort and using an immunohistochemistry method to validate our findings. So the picture are shown on the left of the IHC stains. Brown means the protein is expressed in the tumor. Blue means the cells are there, but there is no protein present. So on the right box plot are laying out the histo score, which is represented by the percentage of tumor cell staining from 0 to 100%, multiplied by the intensity of staining. So 3 being the most intense and 0 being there's no expression. So you can see that in the uncommon ovarian cancers, the expression of ASS1 is slightly decreased. And next slide, please. And I just want to, the previous slide. Thanks. I just want to draw the attention to SCOOT and the in the red box, showing that in the 31 cases that we have, the expression of ASS1 is quite low. Here, I can take her. Sorry about that. She's being a grumpy baby today. Victor has a meeting. Okay, next slide, please. So we go on to show that using a panel of cell lines representing different subtypes of ovarian cancer, that whatever's missing ASS1 is sensitive to ADPEG treatment. So now we wanted to test this in vivo. So to do this, we subcutaneously injected cell lines or patient-derived xenografts of SCOOT in mice. And these tumors are allowed to establish and then randomized to either being treated with saline control or 30 mg per kilogram or 15 mg per kilogram of ADPEG. So these mice are treated weekly, just like how the patients are treated. And as you can see from the growth curve, so the first two growth curve is SCOOT cell lines, and the last one on the right is the patient-derived xenografts. So with saline treatment, you can see that these tumors grow quite fast. So all of them reached human endpoints by about 4 weeks. And when you treat them with ADPEG in the blue and the red lines, the growth of the tumor is significantly decreased. And in some cases, we actually can't find the tumor at the end of the study. So next slide, please. So in conclusion, we find that ASS1 expression is a predictor of, that's okay, response to ADPEG treatment, and that it could be a targeted therapeutic for some of these patients with rare ovarian cancers, especially SCOOT, for which effective therapeutics is not available. And that we're, I think, exploring combination therapies with a conventional chemotherapies should be explored. And thank you very much.
So thank you, Jen, for your work and for the excellent presentation. Yeah, so this was done as part of our team, and Yaman is actually working on a grant to look at these combinations now, which we're doing with Sid Huang from McGill and others. And I should note, Jen is a Vanier Scholar, which is an extremely prestigious award which she got for her work. But this isn't the only thing which has come out in this disease space. We went from a cancer which nobody understood even what it was. Was it a carcinoma or not? But it was called one. And suddenly people got interested in it when the mutation was found. And now there are all sorts of different potential treatments. So immune modulation therapy, which worked anecdotally, but probably not it didn't really work when pressure tested. Combined epigenetic therapy, which we worked on. CDK4 inhibitors, where there was very strong preclinical work from Sid Huang's group. Panitumab, less positive, but it showed an effect. And then, of course, ASS1 targeting, which introduces a really interesting conundrum, where we now suddenly have like nothing to a plethora of potential options to test in the clinic. It's a lethal cancer. We need new options, but how do we process these? So why have we got there with this cancer and with no progress in Sertoli Leydig cell tumors and very little in granulosa cell tumors? And it's because for whatever reason, this cancer is really easy to establish models from. There are 3 excellent cell lines, one of which, COV434, was published as a granulosa cell tumor cell line, but we showed and published in a publication, but it's not, it's one of these. And you can grow PDXs from it. And from a very generous woman who had this cancer last year, she let us grow some of the tumor, which is growing as a xenograft, and the family supported the development that as a model system for sharing. And so it's led to this kind of strange kind of conundrum where we've got, if there's no model system, you can't make progress. But in something like small cell hypercalcemic, which is interesting, so lots of people study it, and there are model systems, suddenly you go from no options to too many options to include in clinical trials. And so we have to find a better way of triaging options. The trial which goes forward shouldn't be based on, who had dinner with who. It should be a really good look at all the options. And this is important in rare cancers because you can only probably run one trial in the world on this cancer at any one time. So moving on to the from the rare cancers into the endometriosis-associated cancers. And there's a lot of our team works on this. And I'm not going to be able to mention the work of all of my students and all the projects ongoing. It's going to be, I'll be connecting a few pieces along a thematic kind of string. So the connection between endometriosis and ovarian cancer was first made in 1925. And the question we're asking right now, and I'll describe how we got there, is to what degree and how does the specific microenvironment shape these two cancers which come from endometriosis, so clear cell and endometrioid? And if we understand it, could this lead to some new treatment opportunities? So both of these cancers are associated with endometriosis, and occasionally you see them both in the same sample. So this is an endometriotic cyst, which has endometriosis, an endometrioid cancer, and a clear cell cancer, all in the same lesion. These cancers are very distinct histologically. No one confuses them. Their behavior is quite distinct as well. With clear cell cancers, particularly once they're metastatic, they don't respond well to platinum taxane and they have much worse outcomes. From a molecular perspective, they have different frequencies of mutations, but there isn't a single mutation class, or mutation signature which is absolutely mutually exclusive between these two cancer types, which may suggest that differences between these two cancers is not what happens along the way. It isn't the mutations, it's actually something else. So most of these mutations are present in endometriosis. You find them in the cancer, you'll find them in the adjacent endometriosis. And we found also that a lot of these mutations can be found in endometriosis that has nothing to do with cancers, such as deep infiltrating endometriosis. And this is a study we did with Paul Young, and Mike and Paul and others are working on this project now. We also found that some mutations like KRAS are actually really common in histologically normal uterine samples. So there's a mistyping here, and not just even in endometriosis, but just in normal uteri, which really begs the question of what are they doing there and what do the mutations mean? So how do these two very different cancers come from endometriosis? And so we don't think mutations provide an answer. So what else could be going on? And here a clue was provided by our work with Greg Moran, where we did a proteomic analysis, and this was led by Melissa McConaughey from my team and Chris Hughes from Greg's. And they developed the SP3 protocol, and we compared proteomic profiles with the different common subtypes. And now the world doesn't need another clear cell cancer biomarker, but we found cystothionine gamma lyase was a protein which was expressed more in clear cell cancers than endometrioid and high-grade serous. And so what we did is we ranked the most differentially expressed proteins, brought in antibodies, but only about 1 in 10 times does an antibody you bring in actually work. And this one worked just splendidly well, so we followed up. So CTH is actually an important metabolic protein. It's involved in the transsulfuration in the cysteine synthesis pathway. And but at the time, we didn't really actually focus on what this did. It was more like viewed as a mark. We found that expression was seen in clear cell cancers and also cell lines, and not in endometrioid and rarely in high-grade serous. It performed really well as a histochemical marker, but you don't really need another marker for this cancer type, but it did perform really, really well. And this was true in the ovary, where it marked clear cell cancers with strong expression, rarely seen in other types. And also as in the rare clear cell of cancers of the uterus, we saw strong expression as well. Where it got really interesting is when we looked at normal tissues. So both in uterus and fallopian tube, we saw this marked occasional cells really, really strongly. We also saw the same thing in endometriosis. And what really surprised me, the cells which marked with this clear cell cancer marker are ciliated cells. You can see the cilia nicely here. I didn't even know there were ciliated cells in endometriosis until we saw this. And there's sort of vague references to them about 40 years ago, and then people lost interest. Now, there are other markers actually of clear cell cancer, which are also seen in ciliated cells, which is curious. And indeed, when we looked at clear cell cancers in more detail, here's a case where we have a cancer, it's CTH positive, a borderline tumor, sometimes called adenofibroma positive, and CTH positive endometriosis. When we look at ER, we see the opposite. It would all be negative here, and this would all be and CTH would be all negative in an endometrioid cascade, but ER would mark the endometriosis and the tumor of adjacent to, and then in the case of endometrioid cancer. So this is what it would be ER expression. And this is CTH expression in endometrioid cancer, negative. ER is really strong, but it's negative in clear cell cancer. And these are mutually exclusive in organoids. And it led to the development of a model. And we published a paper with a really ugly image of Zheng Ji, who's actually a very talented artist, as well as being a physician and scientist, drew this much better figure for us. So the idea was maybe these cancers, clear cell and endometrioid, are different because you have different cells of origin. And could the cell of origin for a clear cell cancer be something like a ciliated cell, or in that trajectory, because ciliated cells are terminally differentiated? This is a model, but all models are wrong, but they can be useful. And this is a famous quote from George Box. We don't think that is what's going on. Other data we have doesn't really support it. So we have to go back to the drawing board. Like, how do you get these two different cancers from the same precursor lesion? So let's think about the microenvironment. So we looked at the cell of origin. We looked at what happened along the way. What about the terrain the cancer has to cross in its journey? This is this toxic mess of an environment, the endometriotic cyst, where it's hypoxic, there are reactive oxygen species, there's iron floating around. It's not a happy place for cells to live. And CTH may actually play a very distinct role in how cells can survive this. And this project is now the first CTH paper was published, led by Dawn, and now Amel, who joined the group and was going to present today, but wasn't able to this week, and we'll show her work at a later date, is leading this project, and we're really excited by it. So his CTH is here, and it's a key part of this pathway which leads to sort of detoxification and enables cells to survive nasty environments. And so what we find is CTH is induced by both hypoxia and reactive oxygen species in clear cell cancer lines, which is really kind of interesting. And if we knock out CTH, the cells don't like to grow anymore. And so in other work, which Amel has led, we've shown that this induction is post-transcriptional. It's really, really rapid. CTH loss decreases the invasive capacity of the cells. So it doesn't only enable them perhaps to survive in an endometriotic cyst, but makes them much more aggressive. And in sarcoma, our colleague Paul Sorensen has shown that CTH loss decreases metastasis. And so this may be very important in the way clear cell cancers behave. And so the question is, could this adaptive response be a viable drug target for clear cell cancer? And this is what Amel, the project Amel is leading. We're really excited by it. And so stepping back, so we've got this endometriotic cyst, and are clear cell cancers and endometrioid cancers manifestations of two distinct means through which mutated endometriotic epithelial cells can survive and thrive in an endometriotic cyst? So, and once you're sort of taken this pathway of survival, it just defines what you are, and you become a clear cell cancer or an endometrioid cancer. And as a rather insane example, so you've got a mob of people here, and if you consider this endometriotic cyst, the Mount Everest of nasty microenvironments, only two people who were quite distinct from each other, a beekeeper from New Zealand and a Sherpa from Tenzing, a Sherpa from Nepal, could make it to the top. Two very distinct individuals, and not everybody. Most of this mob never would have made it over the top. And so it's made us really sort of think about what these cancers are telling us by their phenotypes, by the environments they come from. So how to study this, and this is what most of the team is studying. How do cell context mutation and microenvironment interact to create oncogenic opportunity? So you have a normal cell, you have mutations, microenvironment, and we're using organoid models to study these. So we grow up normal cells, and I want to thank everybody who's been involved in helping us access these cells, all our technologists who've grown the cells for us, and the students who are working on this. we can genetically and chemically manipulate them, and we use single cell genomics to study the outputs. And so we have different students working on different disease types. And then Rodrigo, another student, is working on the specific question of what the microenvironment contributes to the growth of these. And each of these students could tell a really interesting story about their work. But Joyce, who presented at CCCOCR, had a presentation ready to fly. So I asked her to present and tell you what she's doing with some common mutations from low-grade serous cancer. So back to what we're trying to study, it's how mutation, cell context, and microenvironment interact to create the oncogenic opportunities manifest in these very distinct diseases, which we call which we call ovarian cancer, with the hope that if we can understand these, it could lead to novel treatment and prevention strategies. At this point, I'd like to thank my team, and with apologies to all those whose work I didn't specifically reference, but that doesn't mean your work is not valued. And my collaborators, both within OVCare and outside of OVCare, and the foundations who support our work, particularly the BC Cancer Foundation, the VGH Foundation, and UBC Development, as well as the grant funding bodies. And of course, the women who from our community who at the worst moments of their life when they're told they have cancer almost invariably respond with this act of generosity in letting us access their tumor samples to study. So at this point, I'd like to pass the microphone on to Joyce, who's going to tell you about her work using this organoid model to study the origins of low-grade serous cancer.
All right, thanks, David, for such an intriguing talk. I will just quickly go over my project today on modeling low-grade serous ovarian cancers with human fallopian tube organoids and single-cell sequencing. So I, in particular, study the low-grade serous ovarian carcinoma. It's a rare subtype of epithelial tumor, which account for about 5% of all ovarian cancer cases. What we know is that it's slow-growing and less aggressive than its counterpart high-grade serous ovarian cancers. However, it's still characterized with high fatality rates, with only 10 to 20% of women surviving 10 years after diagnosis. And this is primarily due to a lack of early detection strategies and suboptimal response to chemotherapies. So we thought that it would be crucial to understand the molecular events underpinning low-grade serous cancer early tumorigenesis events in order for us to develop more targeted and informed therapeutic options to overall increase patient survival. Unfortunately, there isn't too much we know about this cancer other than the likely self-origin is at the fallopian tube. And in addition, we also know that it harbors a relatively stable genome. And recently, it's been shown that NRAS mutations actually co-occur with EIF1X mutations. And one hurdle in studying this disease is that there is a lack of model systems available. So to date, there is a mouse model taking advantage of KRAS and PTEN knockout. However, the self-origin for that one is ovarian surface epithelial cells. There's also tumor-derived cell lines available. However, neither of these two actually allows us to study the early tumorigenesis events in human. So we thought we want to utilize the novel organoid system. So the organoids, they are stem cell-derived progenitor or cell-derived 3D structures. They can better recapitulate the in vivo phenotypic characteristics than 2D cell lines. So therefore, people are using it to model organ development and diseases. And it's being used in basic research, drug discovery, and personalized medicine. And for my research specifically, we're using it to model disease mechanisms. So the overall objective of my project is to elucidate molecular events underpinning low-grade serous ovarian cancers, specifically how NRAS and EIF1X mutations cooperate to drive the early stages of tumorigenesis. And to do this, we'll be utilizing organoid system and single-cell RNA sequencing technologies. So to start with, we cultured fallopian tube epithelial cells from patients in 2D conditions. And then I introduced mutations lentivirally and tagged each mutation with either EGFP or DS-RED, so we can later resolve them at the single cell sequencing level. From there, we're able to culture fallopian tube organoids, and you can see the non-transduced give rise to not glowing organoids, and the transduced ones, they give either EGFP, DS-RED, or doubly transduced organoids. From there, about 2 weeks, that's the time we allow organoids to establish and allow the transcriptomic changes to happen. We then subject these for histology embedding and single-cell RNA sequencing. Overall, we hypothesized by introducing driver mutations into normal human fallopian tube organoids will lead to transformation events that recapitulate those occurring in the early stages of tumor genesis of low-grade serous cancers. So what are we looking for when we are establishing a new model here? So we are particularly interested in the cytological signs of transformation. So for example, increased nuclear to cytoplasm ratio, nuclear hyperchromasia, prominent nucleoli, cellular or nuclear pleomorphisms. So on the left here, these are fallopian tube organoids, non-transduced. The cells are even in size, distribution and organizations indicating non-malignancy. In the middle, these are organoids transduced with mutations, and we're calling them low-grade serous cancer modeling organoids. There's an overall larger nuclear to cytoplasm ratio. There's also pleomorphism and prominent nucleoli, and a lot of which I've labeled with the colored arrows here. However, one problem with studying low-grade serous cancer is that it's slow growing and a well-differentiated kind of cancer. So any phenotypic or histological differences between normal mutant models would be very subtle. So I thought it would be interesting, a good idea to compare the histology of tumor modeling organoids to tumor-derived organoids. And this is what you see on the right here. We saw a very high resemblance of the tumor modeling organoids and tumor-derived organoids. And again, we saw these signs of these cytological signs of transformation again. So quickly go over the single-cell RNA sequencing workflow. As I alluded to earlier, each mutation is tagged with either EGFP or DS-RED. So at the single-cell level, we are able to decipher which exact cell was untransduced or harbored NRAS or EIF1X mutations, or both mutations at the same time. And from the data itself, I was interested in looking at mutation synergy. So basically, what genes change expressions synergistically in the double mutant versus single mutant organoids. So interestingly, we identify CA125, which is a clinical serum marker for ovarian cancer, however lacks specificity and sensitivity. And two other targets we found was KRT19 and TAC-SDD2. So for KRT19, it's been suggested to be used in conjunction with CA125 as a diagnostic serum marker. And TAC-SDD2 has been found to be overexpressed in serous ovarian cancers, and patient had overexpression of this particular protein had a overall poor survival and shorter progression-free survival than patient with weak expressions. So next, we plan to perform IHC on low-grade serous cancer tissue microarrays, again targeting the genes synergistically upregulated in the double mutant organoids compared to the single mutant organoids. And we plan to validate functionally interesting targets either via genetic, biochemical, or pharmacological approaches. So to quickly summarize, organoid culture and single-cell sequencing can be a powerful tool in studying early tumor genesis events. We're able to establish a new model by introducing co-occurring mutations to normal tissues. Our model can, to a certain extent, recapitulate tumor cytology. And this will be crucial for developing informed, targeted treatment options to improve patient outcomes for women with low-grade serous cancers. In the end, I just want to thank David and my mentor, Dawn, and funding agencies, and especially women who donated their fallopian tube tissues to make all this possible.